All right. Hi, everyone. I'm Noah Snyder-Mackler. I'm a professor in the School of Life Sciences in the Center for Evolution and Medicine at Arizona State, so down in Tempe, not too far from here. I study non-human organisms to try and understand, the how and why we all age differently, which is going to be the title of my talk, right? And I think sort of this setup that we had here already with Representative Schweikert, and then the talk just now as well, mentioned, they both brought up some things that I think are bubbling under the hood. We're all geroscientists, at least most of us probably here are. We're trying to understand mechanisms of aging biology to increase, the years of healthy living, the health span, in particular, right? And but layered on top of all of this is that we are messy organisms as humans, right? We are complex. We live in complex worlds, right? We live in different worlds. We have different access to things, right? We have different health behaviors. We have different behaviors in general, right? All right. So while we're all, we're all getting older, we all know we're aging, right? That's very well, very appreciated. We all age in different ways. That's sort of less appreciated. This substantial heterogeneity in how and why we all age. And this matters, when we're, trying to increase longevity, increase the years of healthy living, uh, because the same sort of mechanistic approach or intervention won't necessarily work for everyone because of this underlying, this bubbling heterogeneity. I think those lights being off would be great, actually. As long as people don't fall asleep, their coffee. Is that bad for you? Oh, never mind. Sorry. All right. Well, no, that's good. All right. So aging is universal. We see it across the tree of life, right? Everything ages in some way, right? Actuarially, right? And it more so. All right, we're going to work through this, and we know that aging is the primary risk factor for pretty much every disease that we see. Right, the incidence of those increase as we get older. This is the premise of geroscience, and Representative Schweikert brought up the fact that could we do things prophylactically? Yeah, that would be great. That's actually what we're trying to do here, right? We want to prevent things from happening. Increase the years of healthy living so that we have a reduced disease burden on society in general, right? And then there's the knock-on economic consequences of what we do with that, very active and healthy, older population. But we'll leave that up to the policymakers for now. Let's think about how we can actually get to that, secondary, maybe champagne problem. Well, we know aging is sort of inevitable, and it happens to everyone, and it happens, and it's the cause of many diseases. What's less appreciated is that we all do this differently, right? And so we age. Some people will, develop certain diseases earlier in life, while others will never develop, a cardiovascular disease. We can think of this as having sort of, different rates in which we age, and different ways in which we age within ourselves in terms of our organs and our cells. This variation here is really the norm and not the exception, right? And so we need to be thinking about this inter-individual and within individual variation in aging when we're trying to come up with these different sorts of interventions. And we really don't, we need to, before we can, intervene, we need to understand what is driving this variation, right? We need to understand mechanistically, how our real world experiences and exposures impact the way in which we age. And there are a few reasons why this is sort of a problem that we're trying to tackle in my lab, and a problem that I think as a field we're trying to tackle, why we don't understand this heterogeneity. One is that we, as humans, live a long time, right? We live across different generations, across different experiences, and so it's really difficult for us to get sort of longitudinal coverage, over the course of a however many year research career, but more importantly, more practically, over the course of a 3 to 5 year grant cycle. We are living in very complex environments, both physically, climatologically, and nutritionally and socially. And so there's this comes down to this fundamental trade-off between sort of very controlled lab experiments and more complex experience, let's say, the controlled lab environment, and then more complex, real world environments, the human experience and exposure and exposome in particular. And now I'm thinking we might actually have the wrong talk here, but we'll see. Yeah, we do. Hang tight. Download the wrong one. Sorry about this. Yeah, there we go. All right. So there's this fundamental trade-off, right? The more realistic your system is, the less control that you have, right? But you're able to capture some of this really relevant and valuable complexity that we see. So I think what we want to do is sort of split the difference, find that sweet spot between balancing this experimental control and this environmental exposure sort of complexity. And I'm going to tell you about two systems in my lab in which we think we find this sort of like Goldilocks split here, and sort of two really probably oversimplified results from some of the work that we've done. The first one is we study this population of macaques, these monkeys that live on a small island off the coast of Puerto Rico. They're a provisioned population, no predators, but so they have some exposures, but not a ton of exposures. They're exposed to the elements. They've got consistent nutrition. We're able to follow them longitudinally. They live on a median lifespan is anywhere from 15 to 18 years, so we can cover at least one generation and multiple generations. And we can capture their complex, daily lives with detailed observations, and then pair that with biological samples that we can collect on an annual basis to try and understand how variation in their environments and what their experiences are and what they're exposed to gets under the skin to sort of mechanistically alter their aging biology. And so the one story I'm going to briefly tell you about is that back in 2017, Hurricane Maria sort of devastated the island, went through the Caribbean, devastated the island of Puerto Rico, and made a direct hit on this island called Cayo Santiago, which you can see here from these satellite images, both before and a month before and a month after Hurricane Maria. It's visually very clear what happened to the island. Almost all of the monkeys survived this hurricane. That's a whole other question that we sort of want to figure out is how, because they don't have shelter on the island or anything like that. That's not really the point of what I'm trying to get at here, is that we're actually, climates are changing broadly, right? And we need to understand sort of what are the impacts of these, extreme natural disaster events, but also things like, exposure to extreme heat. Look, there's been a dramatic reduction in the shade that these animals have on the island, right? That sort of heat exposure is something that is very relevant to those of us living in Maricopa County and in Arizona in general and in the Southwest. Because we've been studying these animals for decades, we have biological samples and other types of data from them. This hurricane presented sort of itself as a natural experiment of trying to understand how this extreme natural disaster might impact the biology of aging in these individuals in a much more controlled way than could be done in humans, where after some of these events, those with means can leave the area, right? And those without the means may not be able to. So there's some sort of selection bias in what you're able to actually capture and measure. And so what we did is we used the blood samples that we collected before and used blood samples that we collected after, did a whole bunch of genomics work on them, and here it's going to be based on transcriptomics. The details are not totally relevant, but what we found was that those animals that had been exposed to the hurricane, when we boil it down to sort of a mark of their biological age in their blood cells, and this is not epigenetic age, it's transcriptomic age, but they're very analogous. So what we found is that those that were exposed to this natural disaster had their ages accelerated by about 2 years relative to their chronological age, right? So the exposure to this hurricane, this natural disaster event, shifted biological age on average by about 2 years. And you'll think, well, 2 years maybe isn't really that much, but remember the fact that these animals live about 3 to 4 times faster than we do, right? So if we do some back of the envelope calculations, this is about a 6 to 8 year age acceleration in humans, if we think of it in human scale. Cool. So that's fairly substantial. We also saw, when we looked at sort of like the mechanistic pathways that were shifting, we saw specific shifts in age-associated inflammation that increased after exposure to the hurricane, and also reduction in some of these other hallmarks of aging, like DNA repair, proteostasis, protein maintenance, and telomere maintenance as well as B-cell proliferation, which is linked to a sort of immunosenescence as well. So some of these phenotypes showing both sort of at a biomarker level, right, that these animals showed accelerated aging, but also it's sort of a mechanistic pathway level in their immune systems that exposure to this environmental stressor accelerated the aging process. Now, another system that we work on is dogs. Everyone here loves dogs, I assume, unless there's some cat people, and I'm sorry, but that's okay. So we do this work as part of the dog aging project, which I'll mention very, very briefly in a second. But we see dogs as also falling in between this sort of control to complexity sort of standpoint. The dogs are very useful models. They're genetic and morphologically diverse. They live in all sorts of environments. They live in the same environments as us, which is, I think, critical here. And they have substantial variation in lifespan. It's the inverse of what we see across animals, where larger organisms tend to live longer. Within dogs, it's very well established that larger animals, larger breeds, live shorter lives on average. Some of the benefits of studying dogs that we can do this at scale is that, one, people love their dogs, so they want to participate in this study to learn more about their dogs and maybe help them live longer. Because they love their dogs, because I love my dogs and others love their dogs, we spend a lot of money on their medical care. They have advanced medical care and lots of detailed medical records on their sort of health history. They share the environment, and not to sort of beat a dead horse here, but they live in a compressed lifespan. So again, we're able to get this longitudinal coverage, and I'm sorry for bringing up horses. And so I'm part of this large NIH-funded, actually from National Institute on Aging, Division of Aging Biology, citizen science or community science project called the Dog Aging Project. It has all dogs. We're capturing survey data on over 50,000 dogs across the US longitudinally. And then I'm going to tell you about a focused subset of these dogs, about 1,000 of them, where we get biological samples from them collected year over year on an annual basis. And my lab focuses on, we lead the epigenomics and transcriptomics side of things, so the functional genomics side of things. And so I'm going to tell you about one quick story where we've looked at the epigenome in these 1,000 dogs year over year to try and understand what are the things that predict or impact variation in the rates of, again, I'm going to reduce it to one number, biological aging. Many of us are aware of DNA methylation as a biomarker of aging. You've heard of epigenetic ages, so I don't need to sort of push through that too much. We developed this epigenetic clock for dogs that was very, very predictive, and actually more predictive than owners or vets when they were guessing their dogs' ages, which is a useful sort of side quest here. More importantly, if you are biologically older than your chronological age, it significantly predicts mortality, and actually an accelerated age increase of about 1 year predicts a 34% increase in mortality, which is pretty substantial. And then one of the most compelling things we can look at in dogs, at least sort of at first blush here, is the fact that there is this size gradient in longevity, and we still don't know what the mechanism is for why larger dogs live shorter lives than smaller dogs. And if we can understand that, it might have implications for understanding inter-individual variation in aging in humans. With this epigenetic clock, we found that giant dogs had a faster rate of epigenetic aging, about 15% faster than these smaller dogs. And I'm at time, right? So I will wrap up. So what I really want to say is, these sort of like two very oversimplified vignettes of these two systems falling where I think is in between, the biological control that we have in experimental laboratory-based systems and the real-world complexity that we actually need to capture and understand if we think these interventions are going to work, in the world, right? In the wild, in humans in general, right? So I'll give you two vignettes where we're actually sort of trying to study this in different ways. I didn't even get into sort of social environmental variation that we see that is very impactful in these two organisms. But I think the take-home message that I want you to all have here is that aging is not just our biology, it's our interaction between our biology and our experiences, our lived experiences, right? And this variation that we see is not a bug in the system, something that we need to control for. It's actually a feature that we need to harness. And with that, I'll thank everyone who's funded and my lab. And yeah, any questions? Time for one question. Yeah. 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 So the question's about sort of like what's the trigger for this accelerate aging after this natural disaster, right? First of all, the second question's easier. The control group is age-matched animals before the hurricane, sampled before, right? We also have within individual measures from before and after where we see similar signatures. What do we think the trigger is? I think the trigger is that you're now, it actually seems like we did the sampling a year after the hurricane. They've been exposed to increased heat for that much more time. And we know that heat exposure and heat stress, even there was a recent paper that came out last year showing that exposure to, days of really, really extreme heat accelerate molecular aging in some way. We think that it's that environmental component that's actually driving this. But I want to emphasize that we've done some other studies here. It's that, I said there's a 2-year age acceleration on average, but that's not everyone. There's heterogeneity in this response, right? And we found that a lot of that heterogeneity is linked to individuals altering their social environments and their social connections, right? So those who increased their social connections a little bit more afterwards were able to have access to more shade at certain points. So this sort of this structural or functional social support that they increased. And we published a paper last year showing that those that did that most actually had better survival outcomes 5 years after the hurricane than those who did not. So yeah. Yeah, so you're basically getting at social gradients, right? Here in terms of stressors and psychosocial stress and things like that. And that's one of the most whatever it is that patterns that leads to that gradient, whether you feel like you are at the top or you feel like you might be at the bottom. That's all that matters in the end, really, right? It's less about what is actually the sort of the fundamental cause of these gradients that you see. Um, and we know from epidemiological studies, population health studies, and animal model studies that exposure to this chronic stress, this unpredictability, is causal to many age-related changes and health-related outcomes that we see.